I study the 5' UTR mutations in prostate cancer. The 5' untranslated regions are part of the non-coding genome, which are largely understudied and form a huge portion of our DNA. And I studied the untranslated regions, which are part of this region, and found that there are mutations that can alter gene expression in prostate cancer. So that forms a repository of resources for finding new therapies for prostate cancer patients. We looked firstly at localized patient DNA available in publicly available datasets in TCGA and ICGC. So we called mutations in these regions, but we also additionally sequenced metastatic castration-resistant prostate cancer genomic DNA from patients enrolled in the University of Washington Rapid Autopsy Program. So we have about 80 patients, and we also called mutations within the 5' untranslated regions of these patients. We compared these somatic mutations and found that they are recurrent within our entire patient cohort, and we analyzed what these mutations did to change transcription and translation. Most of these mutations are single nucleotide variants. A small number are short insertions and deletions. This region could be vulnerable to epigenetic changes, but our data set did not include this. So that could be something that we can look further into. We found a group of them in a metastatic prostate cancer tissues that were mutated within the MAPK signaling pathway. And when we looked at the patient characteristics of these subset of patients, they do present with more bone metastasis at diagnosis, suggesting that there might be some clinical relevance there. So the outcome of this study suggests that post-transcriptional regulation is super important in prostate cancer. So hopefully down the line, we can focus the biology on post-transcriptional mechanisms and have this new area be a source for down the line potential therapies. To personalize these therapies, we can potentially look for mutations within the untranslated regions in prostate cancer to stratify how they can be targeted for therapy. Over the next few years, I would hope that there will be more interest in the post-transcriptional mechanisms of gene expression in cancer, especially in prostate cancer, and to further study the mechanistic functions of what these mutations can potentially do to alter transcription and translation.